Hi, this is Dr. Claire, and today we're going to be talking about animal senses. So animal sensory systems are really important for animals to interact with their environments. It's how they get information about the environment. So for example, a bee can see the color of a flower, and actually flowers may have patterns that are visible to bees that are not visible to other animals that will help the bees to find the nectar in the flower. Bees are actually able to see in the ultraviolet range, and so that allows them to see these patterns that are invisible to other animals like ourselves. Another example, rattlesnakes feed primarily on other mammals, and mammals are generally warm, and so rattlesnakes actually can detect the infrared radiation from mammals in the area, and that helps them to find their prey. And then, of course, some animals have very, very acute senses of smell, like many dogs, that can trace chemicals left behind by potential prey items through the habitat, and that allows them to find prey. And we also use that for, in the case of like bloodhounds, you can use bloodhounds to track people through the forest and help find escaped criminals or lost children, things like that. So a lot of animals have pretty amazing senses that are pretty unique. Now, all senses depend on some sort of sensory receptor. So a sensory receptor is a cell or set of cells that are triggered by environmental stimuli. And they can be broken down to 5 basic categories. So if you have mechanoreceptors. Mechanoreceptors are triggered by physical stimulus. So for example, deformation of the cell. So touch receptors that can detect when you are touched, that's an example of mechanoreceptors. And also the cells that allow us to hear are actually detecting movement of air molecules in the air. So all of those are detecting some sort of movement or pressure changes. We also have pain receptors in our skin, and most animals have these. They can detect things that would be damaging to the animal. So if the tissue is damaged, if there's any kind of tissue damage, it can detect that. Extreme heat, extreme cold, things like that. We also have thermoreceptors. Thermoreceptors can detect heat and cold at levels that are not dangerous. So if you put your hand on a cool item, you can tell that thing feels cool. That's the thermoreceptors that allow you to do that. There's also chemoreceptors. Chemoreceptors detect chemicals in the environment. Those can either be airborne chemicals or waterborne chemicals, and are triggered by those particular chemicals. And then there's electromagnetic receptors that can detect electromagnetic energy, so light waves, heat waves, things like that. Okay, so those are the different types of receptors. So let's take a closer look at how these actually work with a few examples. So in the case of chemoreceptors, there are cells in the body that have particular protein receptors in the membrane of the cell that bind to particular chemicals that might be present in the environment. And so when those receptors bind, then the protein receptor signals a change in the action potential or the polarity of the membrane. So if you remember, the nerve cells tend to be polarized. When this chemical binds, it changes that polarization within the cell, and that can start a signal to be sent to the brain. Okay. Similarly, mechanoreceptors that are detecting some sort of movement or pressure have a similar thing, where a change in the shape of the actual receptor can lead to a change in the polarization of the cell membrane. And things like electromagnetic receptors, and particular photoreceptors, have proteins that are excited by being struck by particular wavelengths of light, and again they change the polarization. In this case, the cells become hyperpolarized. That means that the charge becomes greater across the membrane rather than less. So it's a little bit of a different signal, but it's still the same general pattern, where it's changing the polarization, changing the charge across the membrane of the receptor cell. Okay, so all of these things are working in very similar ways. All right, so let's take a look at how that signal gets translated. So the rate at which these sensory cells get triggered can give you an idea of the intensity of the signal. So for example, if you are lightly touching someone, mechanoreceptors in the skin can detect that touch. And you can tell whether it's a light touch or someone's pushing on your skin, right? And that is interpreted by your brain by the frequency of the signal from that mechanoreceptor. So when it's touched lightly, the signal is sent at a less often, less frequently, at a lower frequency. But if you touch it hard, it has a higher frequency where that signal gets sent, and then your brain interprets that as differences in the intensity of the signal. Same goes for things like bright light, loud sounds. All of these things, the brain is interpreting the frequency of the signal. You also can become acclimated to a signal. So if you have someone who pokes you, you get a very short burst of signal, right? If you have something that touches you for a long period of time, at first you feel that touch, but after a while you become acclimated to it. The signaling rate actually gets slower and slower, and you don't notice it after a while. So if you're somebody who wears, say, an activity monitor, that's touching you all the time, and you probably don't notice it most of the time. But because you become acclimated to its presence, and your nerves are actually responding less than they were when you first put that thing on. Okay. All right. Let's take a look at a few of the senses in more detail. Taste and smell are actually very similar and very interrelated. They are both chemoreception. They are both ways of detecting compounds in our environment. And so smell detects airborne compounds, and taste is detecting compounds that are in water or in oil, generally in our food, right? And actually smell has you have the ability to detect way more compounds with your sense of smell than you can with your sense of taste. In your sense of taste, you basically have 5 different types of receptors. You have sweet, salty, sour, bitter, and umami, or savory. And those are the only things that your taste buds can detect. So if you only combine those 5 tastes, you can't really get the range of flavors that we get from our food. But when you're eating, you are also, little tiny chemicals and particles of your food is getting into your nose, and that's what gives us the rest of our perception of our food. It's the ability to also smell those chemical compounds. And so our sense of smell can detect things like things that smell like vanilla, for example, things that smell like oak, things that smell like vegetal qualities. So we have a whole bunch of different compounds that we can detect with our noses that we can't detect with our tongue. And it's the combination of the signals from the tongue and the signals from the nose that give food its complex taste. A little bit of a personal story. Last fall, I completely lost my sense of smell. I could smell absolutely nothing. I had a virus, or a bacterial infection actually, that attacked my olfactory receptors. And so I could smell absolutely nothing. Zero smell. Nothing smelled like anything. I actually bought deodorant during that time, and it stunk, and I didn't realize it, and because I couldn't smell it at all. But I could still taste, but all I could taste was sweet, salty, umami, sour, and bitter, and that's it. And so, like, I'd eat food, and I'd be like, yep, this is salty. I'd be like, chicken, and I'm like, yep, someone put salt on this chicken. It was really boring. I don't recommend it. So it's a really important interaction that makes your food taste interesting. A lot of animals have ability to detect motion in different ways than we do. For example, fish have what's called a lateral line. It's a line of hair cells that run down the length of the fish, and those cells can detect movement in the water, so they can detect vibrations from other fish. So if you've ever seen fish schooling in a really tight bunch and they all move the same direction, they're actually not looking at each other. They're detecting the movement of the water from the fish nearby and using that as a signal to allow them to move together as a big school. Most invertebrates have what are called statoliths. Statoliths tell invertebrates which way is up. And so the statolith is actually a little tiny grain of sand. It's inside a structure called the statocyst, and it rolls around in there. It's a big hollow ball, and as that statolith rolls around, cells in the wall of the statocyst send signals about where that ball is. So that way, when the ball is in one location, then the animal knows that it's up. If the ball goes to the side, the animal is not facing up anymore. So if you look at, like, say, shrimp, they're usually facing up, and that's how they are able to know, is from these statocyst receptors. Interestingly, if you replace the statolith, the little like pebbly thing inside the statocyst, with something that is magnetic, you can really confuse a shrimp by giving it a magnet that'll cause the statolith to roll to one side, and then it'll think that that direction is down and the other direction is up, and it'll swim sideways. So they can get very confused. Hearing is actually another kind of mechanoreception, and in hearing, what happens is that vibrations in the air are translated into vibrations within a liquid in the inner ear, and then that liquid vibrates hair cells in the ear. So the vibrations in the air are translated across the tympanic membrane through a series of bones to the inner ear. Within the inner ear, there are these hair cells that are able to that are triggered by movement. So as the vibrations in the water move those hair cells slightly against the tectorial membrane, that's how the body is able to detect those vibrations in the air, and then our brain interprets that into sound, which is pretty amazing. Vision is actually a very useful sense. It has evolved multiple times in multiple different animal lineages, and animals have a couple different ways to detect light, which is a type of electromagnetic radiation. Light is electromagnetic radiation within a particular group of wavelengths. So things like flatworms have a very primitive eye called an eye cup, which is basically just some light-sensitive cells that are encased in a pigment layer just on the surface of the skin. So there's no focusing of the light, and they can basically detect movement and shadows, but they can't really see things clearly like we can. Many arthropods have what's called a compound eye. A compound eye has multiple lenses with photoreceptors behind each lens, and so it focuses the light onto these photoreceptors, and but many different lenses on each photoreceptor, which is why it's called a compound eye. Single lens eyes have actually evolved twice, once in the mollusks. So the squids and octopus have a single lens eye, and then again in the vertebrates. So we also have a single lens eye. In our eye, we have a single opening. The light passes through a lens, and it is focused onto the back of the eye, where there are photoreceptors on the retina, and that's how we are able to perceive light. Vertebrates also have color vision, so we're able to see more than one wavelength of light. The way that we do this is that we have two different types of light receptor cells. We have rods and cones. Rods can only detect one wavelength of light, but they are very much more sensitive than cones. So in low light conditions, your rods are more active because they're very sensitive to in low light conditions. In bright light conditions, your rods kind of get overwhelmed and they turn off. But we have cones, and the cones, there's several different types of cones, and each one can detect a different wavelength of light. And so by combining the excitement of the different types of cones, we can perceive color. One thing that I think is kind of cool, humans have 3 different cones, so we can see basically red, green, and blue. So here's the human spectrum. We have these 3 different things. I don't know why the colors aren't corresponding to the colors that they can actually see in. It's actually not quite like this, but you can see that there are these 3 different cones, each one with a different wavelength that they can detect. Birds have a 4th type of cone, and that 4th type of cone can actually detect ultraviolet light. So birds can see many more colors than we can, and I think it would probably be pretty interesting to see the world as a bird. So that's my lecture on animal senses, and I'll catch you next time.